Hi everyone. In this video about sex determination, I want to move on from talking about vertebrates and do a roundup of a variety of other systems. So today we'll be talking about a variety of things. Sex determination in the very well studied fruit fly Drosophila melanogaster, which works by splicing regulation. We'll talk about XXXO systems, which are like XXXY systems, but without a Y chromosome. We'll talk about sex determination by X chromosome elimination, about asexual and sexual generations, about sex determination by allele counting, and then at the end we'll just do a roundup of all kinds of really cool, diverse sex determination systems. So, one thing I want to say before we start out is that in these videos I've been using consistent representations about how we show genes, about how we show regulation. So in this video, in part because I'm using some other people's materials, there are some other ways of representing the same kinds of data. So on the one hand, this is unfortunate because it means that it takes a little bit more time to understand exactly what's being said, but I really think it's worthwhile because this is really good practice for learning to communicate. Different colleagues, different scientists communicate in different ways and use different representations. So getting some practice in learning how to take a new representation and map it onto our understanding is really good. Okay, so let's start out with sex determination in Drosophila. As we did in vertebrates, we can start out with the switch. And so in this case, the switch is just a single gene called sex lethal, which positively regulates itself. Right? So it's an autoregulatory switch. And as we discussed, we expect that to either lead to low levels or high levels of product, not to intermediate levels. In particular, the way that it regulates itself is at the level of splicing. Okay, so this looks a little bit like what we've seen before, but what we see here is what's being shown is two different pathways on the top. That's when sex lethal protein is binding. What we see is that we skip this third exon, but then on the bottom, without sex lethal, we get a joining of the three exons leading to this. So there's something funny here, this stop codon. What that stop codon means is that there's actually a stop codon in the middle of this exon. So if the exon is included in this gene, what this means is that this premature stop codon is actually recognized by the translation machinery, and this transcript is degraded. This is by a process called nonsense-mediated decay, which I hope we'll have a chance to talk about. But for the time being, the important part is that this does not give rise to a protein. Okay, so after we have this switch, the next step is that the sex lethal protein comes and similarly regulates transformer. Look at the similarities here. Again, leading to exclusion, splicing to this point right here, and leading to this exclusion of region 2, giving a functional TRA protein product. In the absence of sex lethal, what we get is splicing in of both 2 and 3. Again, 2 contains this premature stop, and that leads to this product being degraded. So what we see on both cases here is that if we have the splicing regulator, SXL, that gives us the female-specific functional protein coding copy. And if we do not have SXL, we end up with the copy that includes this extra sequence with the premature stop, and therefore we don't end up with protein. Okay, so what does this protein do when it's produced in females? Well, it comes down here and regulates yet another gene, yet again at the level of splicing. And in this case, it's a little bit different. We either splice and our last exon is this exon 3, or we splice and this last exon is exon 5. So we either get 3 plus 4 or 3 plus 5. Okay, so this is a little bit different. In this case, we get 2 different proteins, both of which are transcription factors. We get the DSX, with this variant is associated with females and drives female transcription, and this one is associated with males and drives male-specific transcription. And so we can say that DSX female 1 protein is a transcription factor which turns on female-specific genes and turns off male-specific genes. And then DSX male is a different transcription factor because of alternative splicing. This one turns on male-specific genes and it turns off female-specific genes. Okay, so we've said, as we did with vertebrates, that there's a switch, but how does that switch get flipped? Okay, so Drosophila is an XXXY system, but SXL is on an autosome, sort of as we saw in vertebrates, that the switch genes are not the ones that are on the XY. So how does the switch get flipped? So this has got a few more moving parts. So let's walk through this together. So here we are back to our familiar representation. Here's our SXL gene, and again, it has the 2 different possible products. But we're going to talk about how this gene gets turned on or not. Because remember, once SXL gets turned on, then it will positively regulate itself and continue to be on. And so we just need to figure out how it gets turned on initially. Okay, so I'm using slightly different nomenclature than people sometimes use, but I'm just going to say that this is an X factor binding site. And these XFs are going to be X chromosome encoded transcription factors. That means transcription factors that are encoded by proteins on the X chromosome. Okay, so these protein factors can bind here and positively regulate SXL, but actually they can also bind to these autosome-linked factors, that is, these proteins encoded by genes on the autosomes, and they can't do both. So each XF protein is either going to bind an AF or it's going to bind to this binding site. So what's going to determine which it binds to? Well, what's going to determine which it binds to is basically how much AF there is around. In a diploid individual, we expect to have 2 copies of this AF gene, which are going to give some level of AF protein. So if we have 1 X per cell, that would be if we have a male karyotype, then we're going to have some AF proteins. But those AF proteins are actually all going to end up being bound to the more plentiful, the more numerous AF proteins. I'm sorry, I have a typo here. This should say XF. So this should say XF, and then the AF proteins come and bind to the XF proteins. And so because these green XF proteins are bound to the AF, they won't bind to this and they won't kick this switch into the female state. Right? So if we have 1X, that is a male karyotype, our AF proteins are occupied, our XF proteins are occupied with the yellow AF proteins and therefore will not start our female-specific splicing cascade. So we'll have no binding and SXL will not initially be turned on. Okay, what if we have 2 X's per cell? Okay, so now we have twice as much of these XF protein factors. Again, I'm sorry for the typo. So some of them will be bound to our yellow AF proteins, but these green XF proteins, some of them will still be left over. They will not be bound, and so they'll be able to bind and stimulate SXL, beginning the female-specific switch or female-specific state. So kicking the switch towards females. Okay, so this actually allows us an opportunity to practice some problems. So it's in Drosophila. Drosophila is an amazing lab animal. We can do all kinds of stuff to it. And one thing that we can do is we can generate flies that have sort of funny, atypical sets of chromosomes. So we can generate them with 4 sets of autosomes and 3 X's, or 2 sets of autosomes and 4 X's, or whatever. So for these flies, can we predict what their sex would be? And it turns out that a very good prediction of this is the ratio test. So what we're going to do is we're going to take the number of X chromosomes for a given fly, and we're going to divide it by the number of sets of autosomes. So in a typical male, that would be 2 X's and two sets of autosomes, because it's diploid. So that would be 2 divided by 2 equals 1. And what we find is if this ratio is greater than or equal to 1, that is if it's a typical XXAA, or if we have 3 X's in 3 sets of autosomes, or 3 sets of X's in 2 sets of autosomes, in that case the ratio would be 1.5, that development will proceed down the female route. However, if it's less than one half, so that would be what we'd expect from a single X and 2 sets of autosomes, or 2 X's and 4 sets of autosomes, or 1 X and 3 sets of autosomes, then we'll observe male development. And if it's in between, if it's between 1 half and 1, we'll actually get intersex flies, that is, flies that have some female physical characteristics and some male physical characteristics. Okay, so let's do some of these problems. So what if we had 2 sets of XF, or two X chromosomes, and 4 sets of autosomes? So go ahead and pause it if you'd like a moment to think about it. Okay, so in this case, we're going to have our X to A ratio is 2 divided by 4, which is 1 half, which is what we expect. That predicts that we would see a male phenotype. Okay, what if we had 3 sets of X's and 3 sets of autosomes? Go ahead and pause if you'd like. So in this case, we'll have 3 X's, 3 A's, so 3 divided by 3, so 1, which is indicative of a female physical phenotype. Okay, let's do one more. What if we had 2 sets of X's and 3 sets of autosomes? Go ahead and pause if you'd like. Okay, so in this case, we have 2 X's and 3 sets of autosomes, so 2 divided by 3. So our ratio is 2/3, which is between 1 half and 1. So we would expect intersex flies. So I may have gone through this a little bit quickly. I would encourage you to go back the last couple minutes and work on this until you feel comfortable with it. Okay, so actually we have a more complete pathway here, which is that the AF factors sort of block the binding of the XF factors to the sex lethal. So we now have our 5 major players in the Drosophila sex determination pathway. Okay, so what about the Y here? So as we saw in vertebrates, it's not generally the case that both the X and the Y, or both the W and the Z, are involved in sex determination. Usually it's one of the two, either the X or the Y, plays a dominant role. Let's drill down on that a little bit further. Let's talk about X chromosomes in flies. So X chromosomes in flies have very few genes, like 20 or 30, maybe even, I think it might be 17 in melanogaster, which is actually similar to Y chromosomes in other groups. The genes on this Y chromosome are not part of the core sex determination pathway. So this is unlike in mammals, right? However, genes on the Y do turn out to be important for fertility. XO flies, that is, flies that have an X but no Y, they are viable, they survive, but they're infertile. They don't produce viable sperm. On the other hand, species related to Drosophila melanogaster have entirely lost their Y chromosome. That is, that males are XO. Right? So that's kind of funny to think about, but what it means in meiosis is that the X chromosome actually lines up without a partner during meiosis, and we end up with sperm that either contain X's or contain neither X's nor Y's, because the Y doesn't exist. It might help to understand this a little bit by talking about a very funny and I think extremely cool kind of sex determination in black fungus gnats and in gall midges. So these are 2 groups of flies that seemingly independently have evolved this very intricate system that we'll talk about. Okay, so gall midges are parasites of plants, and they form these galls, these little tumors on the plants. So they induce a kind of tumor, an aberrant growth, on which they then feed. And then the other one are black fungus gnats, so the family is Sciuridae. These are pests that are frequently found in fungus farms, and you can actually often find them in your houseplants. Okay, so in this species, in the adult form, females are XX and males are XO. There is no Y chromosome, as we were just discussing, can be the case. It's thought this is probably kind of like a Drosophila-like molecular switch, but we don't actually know. There hasn't been a lot of molecular biology done on these organisms, although in my lab we're working on it, so stay tuned. Okay, so this is a totally different system, though, and this is so cool I want to talk about it, but I'm a little worried that it's going to unhinge us. So sit tight. So the way that this works is all embryos, all diploid embryos, begin life with 2 chromosomes. Actually, it's even weirder than that, but it makes sense to talk about it as 2 X chromosomes. But some embryos eject one of their X chromosomes from almost all of their cells early in development. So let's draw that out. So we have our gametes here, we have our sperm, that's what I mean by the little tail, and our egg cell. And we have an X chromosome from the father and an X chromosome from the mother, a set of autosomes from the father, and a set of autosomes from the mother. So these fuse to give us our diploid gametes, our diploid zygote, our embryo, with 2 X's and 2 A's. So 2 things can happen. One is all the chromosomes stay put, and we go through fairly normal development to give rise to a female, and then the female undergoes oogenesis to give rise to the next generation of eggs, which have a combination of the red and blue, as we would expect from reassortment, from reshuffling, either X or blue, either red or blue for both X and A. Okay, and then the other possibility is the weirder one. So the other thing that can happen is during embryogenesis, one of the 2 X's, and actually specifically the X that was inherited from the father, gets kicked out. So now we have an adult fly that only has 1 X chromosome, not because it only inherited one, but because it kicked the other one out. So this is how sex determination works in these lineages. It's not what you inherit, it's whether or not an extra X, one of your X's gets kicked out. I think this is super weird and super cool. It actually gets weirder. This is not about sex determination, but let's go ahead and go through it. So notice we've color-coded things here where the red indicates the chromosomes that were inherited through the egg from the mother, and the blue are the ones that were inherited through the sperm from the father. So this individual now still has a combination of maternal and paternally inherited genes, but it turns out when this individual goes to make gametes, to make sperm, that only passes down the maternal genome. Okay, so that's a little bonus thing. That doesn't actually have to do with sex chromosome, and that makes it even harder to keep in our heads. But the main point here is that in these organisms, sex is determined by kicking out an X. And I just wanted to point this out, both because I think it's cool, but also because it makes clear that there kind of can't be a Y chromosome in this system, right? Because everybody starts XX, and then it's about 1 X being kicked out. So, as I said, these organisms are pretty poorly studied. I just wanted to give a shout out to Noel Anderson, who's a student in my lab, who's working on this stuff and trying to understand the X chromosomes and the evolution of this system. And let me tell you one more sort of related funny story in pea aphids. Okay, so in pea aphids, actually, some adult females were produced not by fusion of an egg and a sperm, but actually by asexual reproduction. Okay, so this is a female that will then give rise to another asexual female. So this actually happens by mitosis. This is super weird, and it's not what we're accustomed to thinking about. But it's really interesting to think that nature can be this diverse, right? So this is an animal. It's a bilateral animal like ourselves, but it can give rise to asexual females. So what happens is basically during most of the year, there's this trail of asexual females where this asexual female reproduces asexually, makes a bunch more females, who then also reproduce asexually, and this happens for about 10 generations. Okay, so when the winter is coming, in the autumn, an asexual female will produce again asexually. However, in this case, in some individuals, one of the X chromosomes is eliminated, giving rise to an individual like what we saw, with two sets of autosomes, but only one X chromosome, so XO. That individual will develop as a male. Other individuals will still develop as females. These individuals will produce sperm cells, which interestingly all have X chromosomes. These individuals will develop, will give rise to eggs. And this looks mostly like a normal sexual generation. So we have all these asexual generations, and then occasionally this sexual generation, which is created by eliminating one of the X's to give rise to both males and females. So I think this is super, super cool. Okay, so that's aphids. Let's talk about yet another cool system. So I'm not sure if you've heard this, but the way that sex is determined in hymenopterans, which is a large group that includes bees and wasps and ants, sex is determined not by what genes come in the sperm or what genes come in the eggs, but whether or not there's a sperm involved at all. So females are produced by a fairly normal-looking system where an egg is fertilized by a sperm, giving rise to a fertilized egg. And any fertilized egg, pretty much, will give rise to a female. On the other hand, males are produced from unfertilized eggs. So that would be the case that an egg goes unfertilized, giving rise to an unfertilized egg. And that unfertilized egg will, in fact, develop into a full adult male. So this is sort of the phenomenon that we observe if we study the biology of the organisms without even looking at their molecules. This is what we find, that fertilization leads to females, non-fertilization leads to males. But that still doesn't tell us sort of what happens at the genomic level, right? It doesn't tell us sort of which genes are being involved in flipping this switch. And it turns out that the organism doesn't just simply count the number of genomes. It doesn't directly ask, do I have one or two alleles of each gene? It does something kind of more interesting. So it turns out that in honeybees, sex determination occurs with a single gene, which is called CSD, for complementary sex determination. And what happens with this gene is it has many different alleles. We'll go through this. It has many different alleles, and each one of those alleles makes a slightly different, or actually quite different, protein sequence. And what happens is that if an individual has one allele, that individual will develop as male. But if the individual has two different alleles, the individual will develop as female. Okay, so let's see what that looks like. Let's say that this is our karyotype for bees. So that means that CSD is present on one of these chromosomes. Okay, so if we focus in on that pair of chromosomes, and let's say we collect copies of that orange chromosome from various individuals in the population, and we look at the CSD gene, what we find is all these different alleles. So we're accustomed to sort of calling two alleles, I don't know, maybe gene plus and gene minus, or maybe big A, little a. But in this case, we have so many alleles that we should number them. So we have allele 1, 2, 3, 4, 5, 6. And we have another copy of 4 here, 7, 8, and 9. So we have all these different alleles. And we're just kind of numbering them randomly. So occasionally you'll encounter two like alleles, but for the most part, we find different ones. And so here's a paper where they sequenced CSD gene alleles from a bunch of different honeybee subspecies, And they found a total of 79 different haplotypes, which in this case basically means alleles. So 79 different haplotypes in the sample that they sequenced. Okay, so what happens is that the number of alleles in the embryo determines sex. So if there's only one kind of allele present, then that gives rise to a male. If there are two different kinds of allele present, that gives rise to a female. Okay, so let's imagine we have a haploid individual with just one allele, allele 5. That individual will develop as a male. Or if we have a haploid individual with just allele 17 or with just allele 37, they'll all develop as males. On the other hand, if we have two different alleles, if we have a diploid individual that has one copy of allele 4 and one copy of allele 12, that individual will develop as a female. Or 7 and 19 will be a female. 1 and 23 will be a female. However, if we have a diploid individual that has two of the same alleles, it turns out that that individual will develop as a male. And interestingly, it'll be a male that's, in many species, infertile. So this kind of seems to be wastage, these individuals that are homozygous at the CSD locus, they give rise to infertile males. And again, if we had two copies of allele 11, we would end up with a diploid male. Okay, so that's how the system works, basically how alleles get counted. But what is CSD? Like, what if we look at the CSD function and sequence, what does it turn out that it is? And it turns out to be the case that it's very closely related to the pathway we studied in Drosophila. So CSD is actually a duplicate of this core gene transformer. We could call it like transformer B, but it's been called CSD. So it's been a duplication and then the sequence has changed. So here, let's imagine a diploid where we have two alleles, CSD allele 7 and CSD allele 34. They're giving rise to these related but different proteins. And what turns out to be the case in this species is instead of SXL controlling splicing in transformer, it's CSD that controls splicing in transformer. And so what you find is that if you have two different alleles making two different proteins, then they produce the functional TRA variant, TROF splicing variant. Whereas if you only have one form, it seems to be the case, if you only have one allele, that it does not bind to the transcript, and the transcript gets spliced in this other way, including this extra sequence that has a stop and getting degraded. So this seems to be the way that the switch is kicked in the female direction. In this case, the switch is at the level of transformer, and if you have two alleles, then you'll make functional TRA protein. If you only have one allele, you do not make TRA protein. Okay, so I think that's the number of species that we're going to go through. And so I just thought I would compare what we've done to sort of known diversity of sex-determining genes. This is from the same review that we borrowed the colorful figure with the tree and the different forms of X, Y, and ZW. That is from the tree of sex people. So just to recap, we talked in mammals about the SRY, and in chicken about DMRT1, in frog about DMW, in Medaka about this Y copy of DMRT1 that uses a slightly different name here. We were just calling it DMY, but same thing. In fruit flies about SXL, and then in honeybee about CSD. But I also wanted to point out that in this whole diversity, there are actually 3 more cases where the sex-determining gene is a relative of the genes we've discussed. So in sole, it's DMRT1, just like in several species we studied. And then in housefly, it's this gene called F, which is a paralog of TRA, just like CSD. And in wasp, it's this thing called NV-TRA, which is basically TRA, again, just like CSD. So what we can see in the diversity of known sex-determining mechanisms within animals, we've covered quite a lot of it. And we've also, the other thing I wanted to point out is that we've also covered all the different kinds of sex-determining mechanisms. So remember, a sex-determining Y, that means the Y is what drives maleness. Or a sex-determining W, that's where a gene on the W drives femaleness. Or we could also have a dose-dependent Z, so that's where the number of Zs is what drives things, so a gene on the Z. Or dose-dependent X. And so we can see that we've studied all four of these, and then the fifth one, Haplodiploidy, that's the phenomenon we talked about in honeybees, where males are haploid and females are diploid. Okay, so with our remaining, with the rest of the video, I thought we would just kind of have a romp through some of the other possible types of genotypic sex determination. So these will go a little bit more quickly. Please don't stress out about the details. I just think it's really cool stuff, and I hope you'll enjoy it. So these are the different kinds of genotypic sex determination. This is, again, from the Tree of Sex paper. So we have male heterogamety. That's an XY system. And then we have female heterogamety. That's a ZW system. And then, as we discussed, we have haplodiploidy, where males are haploid and females are diploid. And then we have these 5 other cases, and I thought we would just go through examples for each of them. Okay, so far we've been talking about a case where a single gene determines sex. A single gene kicks the switch in one direction or the other. It turns out to be the case that in some species it's more complicated, that we have multiple different genes that are involved. So what we're looking at here is a species in which actually one pair of autosomes has turned into a ZW system, and one pair of autosomes has turned into an XY system. And so it turns out to be the case, let me remember this, that the W allele, the sex-determining W allele, overrides the Y allele. So that would be this fish right here, where if you have both a W, which should drive femaleness, and a Y, which should drive maleness, that you end up being female. But the main point here is that there are 2 different genes that are contributing to whether an individual develops as male or female. And you can imagine this could really get quite complicated, and I believe it does. But it's just worth keeping in mind that there are systems where it's not a single gene driving sex determination. So another funny case is the UV chromosome system. So a UV is kind of like an XY, but it's in a case where we have a haploid system. So here's the sexual cycle of Ectocarpus, which we mentioned earlier in life history. So during the sexual cycle, basically what we have over here is the diploid. The diploid then undergoes meiosis, which gives rise to 2 different differentiated and multicellular organisms. So this one is the female and this one is the male. And what turns out to be the case is that the diploid has both a U and a V chromosome. But what determines whether a plant develops as a female or a male is whether it inherits the U or the V. So here we have a haploid organism where there is a chromosome that's determining sex, and we call those either the U or the V. All right, moving right along. Another system of genetic sex determination is paternal genome elimination. So this is sort of like haplodiploidy in bees, the one that we went through. So we can start there. Except for, we said before that males came from unfertilized eggs. So in this species, males come from fertilized eggs in the sort of normal sense. So they do have 2 genomes. But kind of bizarrely, they often immediately kick out a single chromosome, a single genome, so that they go from a haploid egg to a diploid egg, and then they immediately kick out one of the genomes, so they go back to being haploid, and they develop as males. So it's basically like haplodiploidy, but with these 2 extra steps, fertilization and then rejection of a genome. In other species, there's kind of a variant of that, where the genome is not actually ejected, but the genome is heterochromatinized, which means basically rolled up into a little tight ball and not expressed and not passed down. And this state gives rise to males. Okay, again, moving right along. There are other systems in which a parasite or endosymbiont, depending, the line between those can be difficult, where its presence determines whether development proceeds as male or female. So let's unpack that. So in these species, there's a endosymbiont called Wolbachia, and that endosymbiont is passed down in some egg cells. So if it is passed down in the egg cells, those individuals are going to lead to a female. They're going to develop as females. So what we're showing here is the nucleus with the two genomes in it and the Wolbachia. However, if the same exact genes, the same exact nuclear genes are in an embryo, but that embryo does not have a Wolbachia, then those individuals will develop as males. So this is a case we call the cytoplasmic gene determination, cytoplasmic sex determination, because the genes that are driving this are not in the nucleus, but they're actually in the cytoplasm in this parasite/endosymbiont. Okay, and finally, the last phenomenon is monogenic broods. So this is a little bit confusing because monogeny is also used for single gene phenomena. So it's kind of confusing. But what it means is that the mother's genotype is what determines the offspring's sex. So basically, within a species, some mothers will give rise to all sons, and other mothers will give rise to all daughters. Okay, congratulations. You made it through our tour of the crazinesses of sex determination, genetic sex determination across nature, in particular in animals. Let's review what we've gone through. So we talked about the splicing regulatory cascade in Drosophila and other insects. And we said that in Drosophila, this switch is flipped by this funny ratio of X chromosomes to autosomes, or more specifically, X chromosomal products to autosomal products. We talked about this new phenomenon where some of these genes are alternatively spliced, with one of the splicing variants having a premature stop codon, which leads to them actually being degraded. So here we have alternative splicing giving rise either to a protein or to nothing. We learned that in some insects, all individuals inherit 2 chromosomes, and sex is determined by whether or not you kick one of those chromosomes out, in which case you're male, or you keep your chromosomes, in which case you're female. We learned about this similar situation in pea aphids, in which we also get males by kicking out an X chromosome, but in which we have alternating generations of asexual females versus a sexual stage with both males and females. The final one that we spent some time on is that in bees, there's a single gene called CSD, and sex is determined by the number of different alleles, whether you have 1 or 2 different alleles of CSG, which is a duplicate of TRA, which is a core SD pathway gene. And then the last bit is that there are many, many cool ways to do sex determination. In conclusion, nature is so cool. Okay, thanks very much.